Hi everyone, welcome back to your channel Agri Clarity, a unit of clarity for sure. In today's Concept Clarity series, we'll be covering an important topic of plant physiology, which is plant growth hormones, most specifically about auxins. In today's session, we'll be covering everything about auxins, like what is its origin, how it's invented or extracted, discovered, uh, by which process it is synthesized, what is its inactivation process, what is its mode of action, everything. So you need not to worry cover anything further after watching this session. So I request every student watching this session to make notes of the everything which I've provided in the today's series. Okay, so let's dive in. So let's discuss what is plant growth regulators. So plant growth regulators are organic compounds that affect growth and development when applied in low concentration. So you have to be attentive that here I have mentioned that plant growth regulators are applied generally in low concentration. Okay, so they can be natural. If they are natural, they are called plant hormones, and when they have their synthetic analogs, they are called plant growth regulators. Okay, so the function of plant growth regulators are they modify life processes or plant structure for improved yield, quality, or harvesting ease. Okay, so let's differentiate about among these three important terms, which is plant hormones, which are plant hormones, plant growth regulators, and plant growth hormones. So plant growth hormone is when the compounds are in its, we can say in its natural form. Okay, natural form. They are occurring naturally, like auxin, gibberellin, cytokinin, ABN, ethylene. These all are plant hormones. When there is also presence of their synthetic analogs, they are called plant growth regulators. Okay, so here is the minute difference. When they have synthetic analogs, they are called plant growth regulators, and when uh, they have exact mode of action and exact structure, I mean their mechanism, but they differ at the molecular level. They are called प्लांट ग्रोथ हार्मोन्स. तो यहाँ से आपको पता चल गया होगा कि प्लांट हार्मोन्स क्या होता है. जब आपको कोई भी कंपाउंड्स नेचुरली मिल रहे हैं प्लांट में, जैसे कि ऑक्सिन, जिबरेलिन, साइटोकाइनिन, एबीए, इथाइलिन, ये सारे क्या हो जाते हैं, हमारे प्लांट हार्मोन्स हो जाते हैं. जब आप इन्हीं के ही ऑक्सिन, जिबरेलिन के सिंथेटिक एनालॉग्स बना दोगे तो आपको क्या मिल जाएगा, उन्हें हम सब उनको कंबाइंडली हम बोलेंगे प्लांट ग्रोथ रेगुलेटर्स. और जब आपके सारे, चाहे वो नेचुरल हो या सिंथेटिक हो, या जनरली कहा जाए नेचुरल की बात की जाती है प्लांट ग्रोथ हार्मोन की अगर हम डिस्कस करें, तो जब आप नेचुरली को मॉलिक्यूलर लेवल पे चेंज कर दे रहे हो बट उसका मोड ऑफ एक्शन सिमिलर रख रहे हो, तो अब आप उस हार्मोन को क्या कहोगे, प्लांट ग्रोथ हार्मोन्स कहोगे. तो इस वजह से इस तरीके से हमने ये तीनों टर्म्स को डिफरेंशिएट कर दिया. सो इट वुड बी नाउ easy for you to eliminate the options. Okay, if there is direct question asking on the definition itself. Next is there are 5 major hormones groups. Okay, so ये सब तो आपको पता होगा. पहला है auxin, second is gibberellin, third is cytokinin, fourth is ethylene, and fifth is inhibitors. Okay, so the nature or origin of auxin is that they are of natural origin, and they also have synthetic origin. The key role is that they are important in cell elongation, apical dominance, and rooting. Okay, so next is our gibberellin. They are of diterpenoids origin, and they have role in stem elongation, breaking dormancy. So you have to be careful that auxin are important for cell elongation, whereas gibberellins are important for stem elongation. Okay, then talking about the cytokinin, they are substituted by adenines, and they promote cell C for cyto, or we can say cells. So they promote cell division and help in delaying the senescence of the plants. Okay, next is ethylene, which is a gaseous hormone. They are helpful in ripening and having apical hook formation. Okay, so next, lastly is the inhibitors. They are of diverse chemical origin. They are generally used for, as the name suggests, they are growth inhibitors, and they are used in stress responses. जैसे for example, if there are plants in the area of drought prominence, or the area which has low temperature, severity or stress, or salt stress, in that cases inhibitors are used for maintaining the growth, reproduction and cycle of the plant. Okay, so inhibitors का ऐसे use किया जाता है. So ये है. Next is let's discuss about the auxin, the growth hormone. So auxin was the first hormone to be discovered. The name was derived from auxin, which means to grow. And अगर हम इसकी बात की जाए, इसको discover किसने किया है? Who discovered it was Cook and Hagen Smith. But isolation was done by Wendt. So you have to be careful that who discovered is the different person, and the isolation was done by different person. So the process which used for the isolation of auxin was a test of phenomenon which is used for the isolation was Avena curvature test. Okay. So as we have discussed earlier, that auxin are of two types, natural and synthetic. Natural, there is only one natural auxin present, which is IAA, which is called indole 3 acetic acid. Okay, सिर्फ एक ही नेचुरल ऑक्सिन प्रेजेंट होता है, और सिंथेटिक में बहुत सारे एग्जांपल्स होते हैं जिसको हमने यहाँ पे टेबुलर फॉर्मेट में बताया हुआ है. सो समटाइम्स what is like, we make auxins in lab and then spray them in what we can say on plants to guide their behavior. So these are called synthetic auxins. So generally synthetic auxins are of much importance because they are generally are of stable and they are widely used in agriculture. Most commonly used synthetic auxins are IBA, NAA, 2,4-D, 2,4,5-T and TIBA. So इन सब के तो आपको chemical name पता होनी ही चाहिए. Okay, you should remember these all synthetic auxins chemical name. So first is IBA, which is indole. Yes, indole butyric acid. Second is NAA, which is naphthalene acetic acid. 2,4-D is known by 2,4-D dichlorophenoxy acetic acid. 2,4,5-T, 2,4,5-T trichlorophenoxic acetic acid, and TIBA is known by 2,3,5 triodobenzoic acid. Okay, so if we talk about the MENA and MCPA, the MENA is known by methyl ester of NAA, and MCPA is known by 2-methyl-4-chlorophenoxy acetic acid. So in this way you have to memorize the synthetic auxins, because there would be a direct question, like they would give you few mixed options of synthetic with natural auxin, and you have the question maybe that you have to identify the synthetic auxins, or you have to auxin which is not which of the following is not a synthetic auxin. So in this way the question can arise from this slide. Okay, so next is types of auxin. As discussed earlier, we have discussed only two type of only the type of auxin based on its origin, which is auxin natural and synthetic. But there is also another type of auxin on the basis of its presence. So we can conclude that type of auxins broadly are classified on the basis of two types. Okay, first is origin and second is presence. Okay, so origin का हमने पहले discuss कर लिया है. Then speaking about the auxins based on presence, first is free auxin. As the name suggests, these auxins are freely moving, or they are mobile, and they are extracted by the use of solvents. And next is bound auxins. As the name suggests, they are attached or bound to something. That something is sugar or amino acids. So if you need to extract the bounded auxins, you have to carry out hydrolysis process. Okay, so free auxins का तो बहुत easy है. जैसे कि आप अगर यहाँ पे मैं आपको एक इन रियल लाइफ एग्जांपल दूं, जैसे ऑयल जो होता है, ऑयल अगर आप मस्टर्ड ऑयल या ग्राउंडनट ऑयल या जो भी ऑयल होते हैं, इन सब का ऑयल का जब आप एक्सट्रैक्शन करते हो तो ये काफी इजीली एक्सट्रैक्ट हो जाता है, क्योंकि जनरली ऑयल में सॉल्वेंट एक डाला जाता है, देन उससे सारे उसके जो भी डर्ट पार्टिकल्स है वो सब निकल जाते हैं, देन आपको एक बहुत प्योरीफाइड ऑयल मिल जाता है. बट बाउंड में क्या होता है, आपका ऑयल के साथ बहुत सारी और चीजें भी होती है। तो अगर आप, मैं यहाँ पे आपको बाउंड ऑक्सिन से समझाऊं तो यहाँ पे ये हो सकता है कि हमारा जो ऑक्सिन प्लांट में प्रेजेंट होता है उसमें ऐसे कहीं कहीं ऐसे पार्टिकल होंगे ऑक्सिन के जो शुगर से अटैच होंगे, कोई एमिनो एसिड से अटैच होंगे। तो शुगर के लिए और एमिनो एसिड्स के लिए अलग अलग प्रोसेस लगेगा उनको निकालने के लिए, है ना। तो इसलिए यहाँ पे बोला गया पहले आपको हाइड्रोलिसिस कराया जाएगा। डिफरेंट स्टेजेस ऑफ हाइड्रोलिसिस होगा। पहले स्टेज के हाइड्रोलिसिस में आपसे ऑक्सिन से आप शुगर को डिटैच करोगे। देन सेकेंड स्टेज ऑफ हाइड्रोलिसिस से आप एमिनो एसिड्स को डिटैच करोगे। देन थर्ड स्टेज में आप सॉल्वेंट्स को इनकॉर्पोरेट करोगे, जो भी रेस्ट ओवर रेसिड्यू डर्ट्स होगा उसको निकालने के लिए। देन आपको एक प्योर फॉर्म ऑफ ऑक्सिन मिल जाएगा। तो इस तरीके से बाउंड ऑक्सिन का प्रोसेस होता है। सो ये डिफरेंस होता है, ठीक है, कैसे ऑक्सिन को एक्सट्रैक्ट किया जाता है प्लांट से। सो दिस वाज इट. So next is key functions of the auxin. Before moving on to functions of auxin, let's I want all students watching this session to imagine something like let's say if you are blowing a balloon, okay, blowing up a balloon, और आप एक balloon को blow कर रहे हो. What if you apply oil on its surface? If you apply oil on its surface, it would stretch more easily, right? So अगर आप ऑयल अप्लाई कर दोगे बलून पे तो वो बहुत आराम से उसमें अगर आप एयर ब्लो करोगे तो बहुत अच्छे से और ब्लो अप होगा. So similarly in this way, auxins soften plant cell walls, okay, allowing them to stretch under internal pressure. That's how plant parts elongate. इस तरीके से auxin help करता है. They help in elongation of plant, okay. So they also you can just by having a little bit common sense of here that if cell or plant is elongating, the number of cells will also multiply. So this would help or boost in gene activity, okay. And with the increased number of cells, the plant would increase its respiration also, and overall these increased number of cells would also have appropriate amount of energy production in themselves. So इस तरीके से what we can conclude from here that they, with the help of auxin, the gene activity is boosted in the plants. Then there is also increase of respiration. Then also there is a appropriate amount of energy production in the plant, okay. So you can see that how auxin is one of the master of the plant growth hormones, okay. So let's now discuss about the key functions. First is cell elongation. It increases the what it does, it increases the plasticity of the cells. For example, when the stem growth happens, it is because of the auxin, okay. As discussed earlier, मैंने आपको पहले बताया था cell elongation is done by auxin, and when we talk about stem elongation, it is done by gibberellin, okay. Second is apical dominance. It suppresses sidebud growth and gives which can be said by having a stronger central growth of the plant. Third is root initiation. It promotes lateral root formation, like when when you conduct a certain amount of cuttings in your plants and you want that they to have a root formation or root initiation in those cuttings, you apply auxin there. So that would help or promote the formation of lateral roots in those cuttings, okay. So next is preventing abscission, or we can say stopping or stagnating the leaves or flower falling from the plants. Then next is inducing parthenocarpy, fruit without fertilization. The most common examples where auxin is used for parthenocarpy is banana, grapes and tomatoes. Next, it stimulates respiration, as I said earlier here, with the increased amount of cells, there is also an increased amount of energy supply and eventually respiration, okay. Next is callus formation. It promotes wound healing and tissue culture. Next is weed control. 2,4-D, which is a synthetic auxin, can be used to kill dicot weeds, okay. So here you have to be careful that it is important for dicot weeds, okay. Yes, for next, lastly is affects flowering or sex flowers. it delays or promotes flowering in some species. So in this way, we have covered every key functions of the auxin. So you have to remember, and you can compare these functions with when we'll cover every इसके बाद जब हम बाकी hormones को cover करेंगे, we'll compare these key functions with themselves also. Okay. Okay. Next is distribution. So, talking about the distribution of auxin, generally auxin are present in shoot tips, young leaves, root tips, and developing buds. So, generally auxin is highest at the tips and decreases as we move down. So that's why apical dominance happens. Okay. So let's see in how much concentration auxin is present among these plant parts. So first is shoot tip. Highest auxin concentration is present there. Talking about the root tip, it also synthesizes, but comparatively less amount of concentration is present there. Then there is coleoptile tip, highest in monocots. So this is very important. There can be question asked here that which of the following crops or species, or we can say crops is appropriate. So which of the following crops has the highest distribution or presence of auxins in their coleoptile tip? So you should know that coleoptile tip is present in monocots. Okay. So in this way you can eliminate the question options here. So next is young leaves. Young leaves has significant concentration, not much, not very less, but a moderate amount. Then developing ex-leaf buds also have a moderate amount of concentration. So in this way we have discussed the distribution of auxins here. Moving on, let's see how these auxins are synthesized in plant. Okay, from tryptophan to IA, this is the process how auxin is synthesized. So there are generally two pathways through which auxin is synthesized. First, in the first pathway, we can see that tryptophan is converted into indole 3-pyruvic acid. Then indole 3-pyruvic acid is converted into indole 3-acetaldehyde, then IA. And in the second pathway, we can see the tryptophan is converted into tryptoamine, then indole 3-acetaldehyde, then IA. Okay. So in this way, you have to memorize, or you have to see, or you have to remember that in how many ways auxin can be synthesized in the plants. Okay. So here I have denoted that destruction and mechanism function of the auxin. So just remember how many ways these two processes happens, because we'll be discussing in detail about these processes in the coming slides. Okay, so destruction happens or inactivation happens in two ways. Mechanism of action happens in 4 ways. Now let's discuss about how auxin is transported in plants. Generally, the direction of transport of auxin is decipetal, or we can say from tip to shoot, tip to base, okay, which is shoot. And when the direction is from base to tip or root, it is called acropetal. So it's important that you should know how auxin is transported. It's in basipetal mode of direction, and the transport is generally in polar transport, means in one direction only. So the transport is conducted with the help of pin-3 proteins, which is also known by efflux. So what is there is, actually the auxin is generally accumulate in cells by the process of simple diffusion. But when it want to transfer or we can say transport auxin from one cell to another, it uses or we can say it uses the help of pin-3 protein here. Okay, so this process is generally known by efflux movement. So ये मतलब होता है efflux movement का, मतलब अगर सेल में जो प्रोड्यूस हो रही है ऑक्सिन वो जनरली सिंपल डिफ्यूजन से प्रोड्यूस हो रही है, और लेकिन जब आप सेल को दूसरे सेल में ट्रांसपोर्ट करना है, ऑक्सिन को दूसरे सेल में ट्रांसपोर्ट करना है, तो वो इफ्लक्स मूवमेंट के थ्रू ट्रांसपोर्ट होती है, और उस इफ्लक्स मूवमेंट की ट्रांसपोर्टेशन में जो हेल्प करती है वो हमारी पिन प्रोटीन होती है. ओके, सो यहाँ पे इसका यही मतलब हुआ. यू हैव टू मेमोराइज दैट the direction of the auxin transport is basipetal, and the transport happens in polar transport with the help of pin3 proteins, and the movement is known by efflux movement. Okay, and these all transportation can be inhibited by the help of TBA and NPF. So this is also important to important here. Okay, so ये सारे आपको पता होना चाहिए. हमको तीन चीजें ही हैं यहाँ पे. बेसिपेटल पोलर ट्रांसपोर्ट, the movement is efflux movement with the aid of pin3 proteins, and these all transportation is inhibited by TBA and NPF. Okay, moving on. Yes, I want to discuss here this diagram. When every time when the auxin is discussed, phototropism comes along. That means जब भी हम auxin का discussion करेंगे तो वहाँ पे एक term phototropism हमेशा आएगा, because as we know that auxin is produced generally on the apical meristem or meristem of the shoot. Okay, so ये जो accumulated molecules auxin के हैं, generally, or we can see, I know some of you have seen that some species or some crops, they generally turn towards the or grow faster or at the much faster rate when they are turned around to the direction of sunlight. Okay, so यही होता है auxin का. जब when there is higher concentration of auxin present in the meristem, the plant turns to the direction of the sun direction. Okay, towards the direction of sun. ठीक है, so इस phenomenon को क्या बोलते हैं? जब आपका कोई plant turn हो रहा है with the towards the direction of sunlight or the shade light side, so this phenomenon is called by phototropism. So आपको ये पता होना चाहिए कि auxin के साथ phototropism term भी साथ साथ चलता है, क्योंकि जिस plant में या जिस species में सबसे ज़्यादा highest amount में अगर auxin produce हो रहा है, वो plant for sure phototropism show करेगी. Okay, so this is the generally सारे plants पे auxin होता है, तो इसी वजह से जो plants होते हैं they are generally fuller in the time of the daylight and they are generally shrinker in the time of the night time. Okay, so ये phenomenon होता है auxin का phototropism में. Okay, next is auxin inactivation and mechanism of action. I have discussed this earlier, that inactivation happens in 2 ways and mechanism of action happens in, or mode of action happens in 4 ways. ठीक है? So the auxin inactivation happens in 2 ways. The first way is by the help of enzyme and second is by the help of light. In enzyme, the IA oxidase, which is the enzyme, helps in breaking down the auxin in the presence of oxygen and produces CO2 and water. Okay, and in the light or by the light UV, or we can say with the help of UV or gamma rays, we can degrade auxin. Okay, and this is also known by photooxidation process. Okay, so हम कभी-कभार क्या होता है, हमें auxin को inactivate करना होता है for certain purposes. तो आप ये दोनों तरीके से आप उस प्लांट में ऑक्सिन को इनएक्टिवेट कर सकते हो, IS ऑक्सीडेज एंजाइम की हेल्प की वजह से, या फिर बाय द यूज ऑफ यूवी और गामा रे लाइट. ओके, नेक्स्ट इज मैकेनिज्म ऑफ एक्शन. सो हाउ ऑक्सिन have a or react in a plant, or how it conducts its mode of action, is that first it increases the wall plasticity. As we have discussed earlier, कि जैसे जैसे प्लांट में सेल का नंबर बढ़ते जाता है तो और उसकी बढ़ती जाती है और इंटरनल प्रेशर बढ़ता है, तब आपके उस टाइम पे ऑक्सिन हेल्प्स इन स्मूथिंग द प्रोसेस. ओके, सो दिस वाज इट. Second is it stimulates gene expression for proteins. What means that they actually enhances the mRNA synthesis, which helps making the protein for cell expansion and eventually stimulating the gene expression of the plant. Okay. Next is they auxin boost in the respiration and ethylene. as we have discussed earlier, how boost in respiration happens, like when with the increase in cells, the cells will have an increased amount of respiration. They will have or they will support much amount of energy production in them. So that's how Auxin helps in boosting respiration. And talking about the ethylene here, Auxin actually triggers the ethylene and it helps in modifying the plant growth. So that's why it's important, or we can say how Auxin also helps in conducting the more, mechanism of or help in modifying the plant growth with the help of Auxin. Okay, and eventually leads to cell expansion we have discussed earlier. So in this four-way mechanism of or we can say mode of action of Auxin happens. Okay, so this was it. Let's before the final wrap up, let's quickly revise important points which we have discussed in today's session. So first is Auxin is the growth master hormone, or we can say master regulator of growth. It works in tiny doses, but effects are very huge. The roles are that it helps in elongation, rooting, parthenocarpy, and weed control. Weed control in mostly in dicot weeds. Then its transport is polar, which we have discussed that the polar transport is done with the help of pin-3 proteins, and that movement is called efflux movement. Then they are made from tryptophan. Generally, they are two pathways we have discussed earlier. Then you can destroy or inactivate the Auxin by help of enzymes and light. Enzymes may we have discussed IA oxidase, and light we have discussed UV rays and gamma rays. So in this way we have recapped all we have learned today. So I have come up with real life applications where Auxins are generally, or we can say frequently used. First is that propagating plants via cuttings. We have discussed in the key function table that how Auxin can help in promoting lateral root in the cuttings. Then next is inducing flowering in pineapples. Then next is delaying the fruit drop in apples. Then next is creating seedlessness in fruits. The important examples were banana, tomato and grapes. Okay, and lastly is the controlling weeds in like wheat fields. Okay, so this was it. Let's practice few questions based on today's session. Let's see how many students have actually understood today's topic. So please answer these questions and drop the answers in the comments below. I'll see how many of you have how many of you have understood this topic very well. So first question is, which of the following is naturally occurring Auxin in plants? First is NAA, next is IBA, third is IAA, fourth is 2,4-D, and lastly is cytokinin. Second question is, which of the following functions is primarily associated with Auxins? First is breaking seed dormancy, second is inhibiting stem elongation, third is promoting apical dominance, fourth is stimulating stomatal closure, fifth is delaying senescence. Third question says, Auxins are used in agriculture for, first is breaking seed dormancy, second is enhancing fruit ripening, third is weed control in cereal crops, fourth is increasing leaf senescence, and fifth is closing stomata. The fourth question says that what is the primary transport mechanism of Auxin in plants? First is active transport in both directions, third is passive diffusion, second is passive diffusion, third is polar transport from shoot to root, fourth is transport via xylem only, and fifth is non-polar movement through phloem. Fifth question says that which of the following is not a typical function of Auxins? First is root initiation in cuttings, second is prevention of fruit drop, third is delay in flowering, fourth is cell elongation, and fifth is phototropism. Then last question is application of Auxins on one side of the stem causes. We have discussed this with the help of picture. So I guess you know the answer. First is option says that no change in growth. Second is equal elongation on both sides. Third is growth inhibition on the treated side. Fourth is bending of stem away from the light, and last is bending of stem towards the light. So this was it. So you can, apart from these, you can say that in your exam questions may not ask what is auxin, but how it behaves under different conditions. So questions can be based on that. So focus on its role, movement and application in agriculture. Okay. So just like humans have hormones that control growth and emotions, plants too have their own chemical messengers. Auxin are the leaders of this pack, making sure that the plant grows smart and not just tall. So this was it. Today we have covered auxin. We will come up with the next session of gibberellins with such detailed content. So make sure you are subscribing to our channel to get all the notification of the upcoming sessions. Also, you can check our AFO test series 24/7, which is available on our website Clarity for Sure, or C4S courses. So we have made test series of exactly same level and exact same pattern of the AFO mains, and we have divided each subject of the AFO in blocks, and so far we have conducted more than 60 plus MCQs on the daily basis. So you can check our courses, and also we have provided crisp short notes and revision notes, or we can say master notes for our subjects as well. So please check out our sample notes which are present on, or we can say test series which are present on our website, and do check out. We are also having detailed full length test conducted every Sunday and Saturday. We have current affairs tests being conducted on a website. So please check out. You can also join our Agri Clarity Telegram channel for more updates on upcoming news or agri related information. That's all for today. Thank you and have a nice day.